What is TA plasmid? TA plasmid is the tumor-inducing plasmid. TA plasmid is used as a vector for Agrobacterium-mediated plant transformation. Using restriction enzymes, plasmid can be linearized and a gene of interest can be ligated. The recombinant plasmid can then be reintroduced into the Agrobacterium by a process called transformation. Once the Agrobacterium infects the plant cells, the plasmid gets integrated into the plant genome and replicates during cell division. These are the general features that the TA plasmid carries a segment of DNA called the tDNA that integrates into the plant genome. The tDNA region of the TA plasmid carries genes responsible for inducing tumors in plants. The foreign DNA of interest can be cloned into the T region of the plasmid, and improved plant traits can be obtained, like disease resistance, salt tolerance, etc. The Ti plasmid has all genes for inducing tumor. It also has virulence genes. The vir genes are responsible for excision, transfer, and integration of tDNA into the plant genome. These are the basic elements of Ti plasmid. It has a tDNA border sequence, a virulence region, tDNA region, and opine catabolism region. So you can see here the tDNA region is flanked by the border sequences, left border and right border, and there comes the tDNA region. Those are responsible for producing growth hormones as well as opine. And the Ti plasmid also has an opine catabolism region where the opine produced from the T region can be metabolized using the enzymes produced from the opine catabolism region. And it also has a virulence region that is responsible for transferring the tDNA into the bacterial genome. So coming to the tDNA border sequence, the tDNA region is responsible for inducing tumor in plants, and the tumor formation is introduced by producing plant growth hormones for the proliferation of the transformed plant cells. The tDNA region is flanked by 24 base pair direct repeat sequence called the border sequence. The role of 24 base pair repeat sequence is for the transfer of tDNA into the plant cell. So you can see here the left and right border here. So these are the 24 base pair repeat sequences. Those are involved in transferring the tDNA into the plant cell. And the other region is the virulence region, or each has vir genes, or the virulence genes that are responsible for creating virulence or inducing or excision and transport of the tDNA region into the plant genome. So the vir region on the Ti plasmid is a collection of genes whose collective function is to excise the tDNA region of the plasmid and promote its transfer and integration into the plant genome. Phenolic compounds such as acetylsyringone activate the vir-A gene, which is constitutively expressed transmembrane protein. The activated vir-A gene acts as a kinase that is phosphorylating the virgin gene. In its phosphorylated form, the Virg acts as an activator of transcription for the remaining virgin operons. So it's a cascade of events that will happen from the virulent region. Once the phenolic compounds are induced, it will activate the Vir-A gene, and that further act as a kinase for phosphorylating the another gene called the Virg gene. And once it is phosphorylated, Virg gene act as an activator of transcription for the remaining virulent genes. The Vir D1 plus D2 together have endonuclease activity and make single-stranded cuts within the left and right borders. The Vir-E acts as a single-stranded DNA binding protein, protecting the single-stranded tDNA region during the transport phase of the process. Once inside the plant cell, the complementary strand of the tDNA is synthesized. So these are the roles of virulence region of the Ti plasmid. So they are mainly involved. It's actually a cascade of events happening when there is a signal from the phenolic compounds. So it's a cascade of events or the signal paths that activates one after the other, and making the tDNA region excised from the plasmid and transported into the plant genome. The tDNA region is flanked by direct repeat sequence called the border sequences, and the tDNA region gets incorporated in the plant genome and is responsible for inducing tumor. The tDNA region has genes for producing plant growth hormones like auxin, cytokines, and opines. Opines are nothing but specialized amino acids. So these are specialized amino acids that bacteria can catabolize. Only the Agrobacterium can catabolize these kind of specialized amino acids. That is the specialty of this amino acid. Opine catabolism region is responsible for catabolizing the specialized amino acids produced from the tDNA region. Based on the opine produced, Ti plasmids are classified into two types. They are napalline type and the octopine type. Opines act as a nitrogenous source for Agrobacterium tumefaciens. There are a variety of uses for the Ti plasmid. It is widely used in plant genetic engineering. It is also used for transfer of genes in plants and also for development of stress-tolerant plant varieties. Thank you for watching. If you like the video, please subscribe.